Today's topic is proteins. In today's class, I'm going to tell about the introduction of proteins, physico-chemical properties of proteins and amino acids, different structure of proteins, and finally the classification of proteins and amino acids. Proteins are most abundant organic molecules of living system, so which is consisting around 50% of dry weight of cell, and these proteins can be synthesized by the process of translocation, and these proteins will be useful for structuring and functioning of the cell, for regulating body tissues and organs. And proteins can be hydrolyzed and forming amino acids. We are having 20 amino acid in animals and plants and microbials. Collagen is the most abundant protein which is present in animal. And properties of proteins. So proteins have the solubility, so which is producing a colloidal solutions instead of true solutions in water. And these proteins are having higher molecular weights. For example, insulin is having 5,700 Dalton units, and hemoglobin is having the molecular weight of 64,450. And the shape of the proteins. Here, the proteins are having variable shapes, like in insulin, so it is globular shape. In albumin, the protein is oval shape, and in fibrinogen, so it is elongated or fibrous shape. And the isoelectric pH of proteins, here proteins are producing deuterions or dipole ions due to presence of N terminal and C terminal. N terminal is mainly due to presence of amine group, and C terminal, which is mainly due to presence of carboxylic group. Amine group is producing a basic nature, and carboxylic group is producing acidic nature. So due to presence of these two ions, here it is producing deuter nature. For example, hemoglobin is having the pH of 6.7. So this is about the properties of proteins. And next one is amino acids. The name itself indicating that amino acid is containing amine group and acid group. So this is the basic structure of amino acid. Here amine group is present, so which is responsible for basic nature, and here acid group is present, which is responsible for acidic nature. Here alkyl side chain is also present. The alkyl side chain is which is replaced by hydrogen, then this amino group is called to be glycine. If this R is replaced by methyl group, then it is called to be alanine. If this R is replaced by methanol group, and which is called to be a serine. And these amino acids are having the basic elements of carbon, hydrogen, oxygen, and nitrogen. But in few amino acids are also having sulfur group. For example, cysteine is having sulfur group in the basic amino acid structure. And these amino acids are producing physical properties like solubility, melting point, taste, optical isomerism. So these amino acids are water soluble and producing more than 200 degrees melting point. And some amino acids are sweet in taste, and some are tasteless or bitter taste. And amino acids are producing optical isomerism. So why? Because here there is asymmetric carbon which is present in the amino acid. Due to presence of asymmetric carbon, here it is producing optical isomerism. And these amino acids are also producing ampholytic solutions. Why? Because due to presence of two types of amino acids like basic amines and acidic carboxylic group, so which are present in the same amino acid. So that's why this is called to be ampholytic. And these amino acids are also producing a chemical nature, like it is reactive with alcohols and producing salts. And these amino acids also reacting with amine group and forming amides. And it is also undergoing decarboxylation reaction, and it is giving amines. And here these amino acids can be differentiated into 7 groups based on chemical nature. So these are the seven important chemical natures like neutral nature, acidic, basic, sulfur containing, alcoholic, aromatic, heterocyclic. In neutral nature amino acids, so which are containing one amino group and one carboxyl group. So as I told already that amino group is responsible for basic nature and carboxyl group is responsible for acidic nature. So these are the examples for neutral amino acids: glycine, alanine, valine, leucine, and isoleucine. Acidic amine groups are having one extra carboxyl group. Basic amine groups, basic amino acids are containing one extra amine group. And sulfur containing amino acids have sulfur, like cysteine, as I told you already that cysteine is having one sulfur group. Alcoholic, so amino groups are having hydroxyl group. Some amino acids are having aromatic rings and heterocyclic rings. So these are the different type of amino acids: neutral amino acids, acidic, basic, alcoholic, sulfur, and aromatic. So here neutral nature so will be mainly due to presence of carboxylic acid group as well as amine group. So aspartic acid is producing acidic nature due to presence of extra carboxylic acid group. Here two carboxylic acid groups are present. Lysine is producing basic nature. So why? Because it is having extra amine group. Serine is producing alcoholic amino group. Why? Because it is producing alcoholic functional group. Cysteine is having a sulfur. So that's why this is sulfur containing amino acid. And phenylalanine is called to be aromatic. Why? Because it is having aromatic ring. Peptide formation. Here amino acids will be bind each other by the peptide linkage. This peptide linkage is mainly due to condensation reaction. That means here water molecules will be removed from these two amino acids and there is a peptide linkage will be forming between these two amino acids. Here two amino acids are involved in this reaction. So this is called to be dipeptide. If many amino acids are involved in the chain formation, then it is called to be polypeptide chain. And the next one is structure of proteins. Here the proteins are having four basic structures: primary, secondary, tertiary, and quaternary. The primary structure, which is having a linear sequence of amino acids. So this is a linear sequence, and which is useful for the formation of backbone of proteins. Here this linear sequence will be bound by two important bonds. So one is peptide bond, so which is forming in between the amino acids, and the second one is disulfide bond, so which is present in between two side chains. So this is the side chain, this is another chain. So here disulfide bond is forming in between these two side chains. Here in this primary structure, it is having two terminals. This is left hand side we are having N terminal and right hand side we are having C terminal. So N terminal is mainly due to presence of amine group at one end, and C terminal is mainly due to presence of carboxylic group at another end. So these are the important functions of primary structure, so which predicts the secondary and tertiary structures. So primary structure, which is having the genetic sequence, an amino acid sequence, so which is useful for the predicting of secondary and tertiary structures of proteins. And these primary structures will also be useful for identifying genetic diseases. For example, sickle cell anemia is a genetic disease. So there is a change in the sequence of amino acid. It is mainly due to some important amino acids which are present in the genetic sequence. So if you change the genetic sequence, that leads to the sickle cell anemia. And we can understand the molecular mechanism of proteins, the folding of the proteins, the arrangement of the protein, the size of the proteins. So we can get the information, we can understand these concepts by the structure of primary proteins. And also we can trace the evolutionary paths. And these primary structures can be determined by two important techniques, Edmond degradation and gene sequencing method. Secondary protein structures. So these secondary protein structures are having a specific coiled structures, a linear chain along with specific coiled structures. Here it is having three types of secondary structures: alpha helix, beta pleated sheets, and collagen helix. So this is called to be alpha helix. This is beta pleated sheets. The third one is collagen helix. In alpha helix, the secondary structure, so which is in spiral nature, so which is coiled and tightly packed. Here, while it is spiraling, there is instability is produced in between these amino acids. To get the stability, here there is a hydrogen bond will be forming to stabilize this helix. Here 3.6 amino acids which are present in each turn in these alpha helix structures, and length between this point to here will be 5.4 angstroms. And these alpha helix structures which are present in globular proteins, for example myoglobins and troponins. And next one is beta. In this structure here, so these linear primary structures will be aligned side by side, and here it is forming a beta strand. So this beta strand which is having 3 to 10 amino acids, and here also there is instability is produced. So that's why here a hydrogen bond will be produced in between these two sheets, and these hydrogen bonds will be two types: parallel and anti-parallel type. In parallel hydrogen bonds, here N terminals will be in one side and C terminals will be in another side. That means the direction of hydrogen bonding will be in one direction. But whereas in case of anti-parallel type, here, so these two directions are anti-parallel in nature. So this is the anti-parallel type of sheet. So why? Because here, so N terminal is present here and C terminal is here. And again, this is C terminal and this is N terminal, so which are anti-parallel in nature. And the third one is tertiary structures. So which is also called to be three-dimensional conformations. So these tertiary structures which are having alpha helix structures and beta pleated sheets, and these three-dimensional tertiary structures which are useful for the biological functions of the proteins. It is maintain the integrity of the proteins, it is maintain the size and shape of the proteins, three-dimensional shape. And it is also useful for the evolutionary origins, for finding the evolutionary origins by these tertiary structures. If we change the size or shape of these tertiary structures, the activity of this protein will be altered. Domains. Here these tertiary structures are available in the form of domains. Here in this picture we are having two domains. This is one domain and these two domain. And these domains are made up of globular clusters. So these are the globular clusters are all together and forming a domain. In each domain, there will be 200 amino acid residues will be present. And this is the example of for domain. So this is the two domain protein, that is glyceraldehyde 3-phosphate dehydrogenase. And due to presence of a size, a structure, here it will be useful for binding the small molecules on this enzyme site. And these domains generally independent. So why? Because so which are present a small globular proteins. Generally these small globular proteins which are water soluble. And these tertiary structures, so which are get stabilized by these important bonds, like hydrophobic bonds, disulfide bonds, ionic bonds, and hydrogen bonds. Due to presence of these bonds, these interactions, here this tertiary structure is getting stability. And this tertiary structure can be determined by two important analytical techniques. First one is X-ray crystallographic technique, and second one is nuclear magnetic resonance technique, so which is also called to be NMR technique. And the fourth structure is quaternary structure. Here, this is the quaternary structure of protein, so which is having multiple polypeptide chains. Here, all multiple polypeptide chains are held together by non-covalent bonds, non-covalent interactions. For example, hemoglobin. Hemoglobin is the example for the quaternary structure, which is having two alpha chains and two beta chains, so which are held together by non-covalent interactions. So these are the structures of proteins. So this is the primary structure, so which is having the amino acid sequence, which is in linear nature. So this is the secondary structure. So secondary structure is having coiling nature. So this is the alpha helix coil, and this is pleated, beta pleated sheets. So this is comes under secondary structures. And these secondary structures will be present in three-dimensional picture, then it is called to be a tertiary structure. Here also we are having alpha helix structures and beta pleated sheets. And the last one is quaternary structure, so which is having multiple polypeptide chains. So these are the multiple polypeptide chains which are held together and forming quaternary structures. And the next classification of proteins. Here proteins are classified into three important groups functionally. So it is mainly depending upon functions, chemical nature, and solubility, and nutritional importance. The first one is functional classification. The first one is structural proteins, example keratin and collagen. So these structural proteins, so which are fibrous and stringy nature, so which are giving a mechanical support, so which is most abundant in the nature. And the second one is enzymatic proteins, which is producing enzymatic reactions, example pepsin. Transport proteins, which will be useful for the transportation process, example hemoglobin. Hemoglobin is useful for the transport of oxygen. Hormonal proteins, so which is useful for the producing hormones, insulin, growth hormones. Contractile proteins, so which is present in muscle cells in the form of actins and myosins. Storage proteins, so which are present in plant seeds and egg white. So these storage proteins are mainly storing the metal ions and amino acids. Example, albumin. Genetic proteins, examples nucleoproteins, which are, these genetic proteins are containing one or more long chain of amino acids, which will be useful for DNA replication and gene expressions. And the defense proteins, which provides a defense to the body, immunoglobulins. Receptor proteins, so which are found in cell membrane, and it will be useful for the binding of specific substances outside the cell, example hormones and viruses. For hormones and viruses, it will be useful. And classification based on chemical nature and solubility. So here three important types: simple proteins, conjugated proteins, derivative proteins. A simple protein which is having amino acid sequence, and conjugated proteins which is having amino acid sequence along with non-protein phosphatic group. Here the non-protein phosphatic group, which is tightly bound with the protein group. So generally this non-protein phosphatic group may be organic or inorganic type. Organic phosphatic groups which are present in vitamins, sugars, and lipids, and inorganic prosthetic groups, which are present in metal ions. The third one is derived proteins, so which are derived from the simple proteins and conjugated proteins. So these derived proteins are denatured proteins or degraded proteins. So proteins are classified into three important types: simple proteins, conjugated proteins, derived proteins. Simple again converting into two important types: globular proteins, fibrous proteins. So these are the examples of globular proteins: albumin, globulin, histones, and fibrous proteins: collagen, elastin, keratin. Conjugated protein, here it is having a conjugated part, that means a protein and non-protein part. And here in lipoproteins, here lipid part, so which is a non-conjugated part, so which is which is non-conjugated part. Here it is present, non-protein part. Here in phosphoprotein, phosphate ions which are present, so which are non-protein part. Metalloprotein, metal ions are present in non-conjugated part. The third one is derived proteins, which are derived from the simple proteins and conjugated proteins, which are two types: primary and secondary proteins. So these are the examples of primary and secondary proteins. And the finally summary part. So proteins are most abundant organic molecules. So they perform structural and dynamic functions in the organisms. It can be useful for the construction of the cell membrane and also useful for the metabolic reactions. And these proteins are made up of polymers of amino acids. So that's why these are large or macromolecules which are present in the cell. Here amino acids, so which are containing amine group and carboxyl group. So we are having 20 amino acids in our body. Now we are having four basic protein structures: primary, secondary, tertiary, and quaternary. Primary is a linear basic structure. Secondary is twisted. Tertiary is three-dimensional structure. And quaternary, so which is having different polypeptide subunits and held together by a non-covalent bonds. And proteins are classified into three important types: simple proteins, albumins, conjugated proteins, so which is having protein part and non-protein part, example glycoproteins. And the third one is derived proteins. So these derived proteins are derived from the simple proteins as well as conjugated proteins. This is about proteins, so which are one of the important biomolecules present in cell. Thank you.